Okay, so hello. Today we're going to be talking about phylogenomics. So we are going to be looking at how the trees that we've been looking at so far, which are associated with duplications, losses, divergence, etc., can be now weaved into species trees and also weaved into coalescent trees, and how we can use that information to infer gene duplications and loss, as well as deep coalescence events. So basically the point of the lecture is that it's going to be trees evolving inside other trees. And this is the last lecture before the quiz. I mean, we have one more lecture on single-cell genomics on Thursday, and then the quiz will be next Tuesday. But it's the last lecture that will be part of the quiz, and it completes the first 5 modules of the course before we get into the research directions. So today we're wrapping up the section on comparative genomics, on genome scale evolution, on phylogenetics, and today phylogenomics. So the goal is going to be to actually distinguish between gene trees and species trees, to talk about reconciliation of gene trees and species trees, and also coalescence of duplication and loss in the context of population differences. So first, we're going to be talking about reconciliation. How do we map gene trees to species trees? How do we infer orthologs, paralogs, gene duplication and loss using this mapping? And then the problem of reconciliation with specific examples, and also maximum parsimony reconciliation. Then we're going to be talking about how we can in fact improve on gene tree reconstruction in the context of the species tree by using the whole genome as a training set. This is very similar to the evolutionary signatures that we've been talking about, where we can actually learn how things evolve from the whole genome, and then use what we've learned in the context of any one locus to increase the clarity with which we can infer these signals. So first, we're going to be looking at how we can leverage many gene trees simultaneously across the entire genome to build species trees using either supermatrix, supertree, or minimum parsimony cost methods. And we're going to be looking at how, after we've built a robust species tree using genome-wide information, we can then go in and build better gene trees by scaling the trees, by learning rates, and by building a generative probabilistic model of gene evolution. Then we're going to be looking at evolution in populations, basically models for population level evolution and algorithms. We're going to look at the Wright-Fisher model, which is moving forward in time, and then the coalescent model, which is moving backward in time, and then multi-species coalescence. And then we're going to study coalescent times distributions and coalescent simulations that can be inferred using these models. And then we're going to talk about incomplete lineage sorting. We're going to blend population and species models to basically use incomplete lineage sorting for species tree, divergence times, population size, and how to deal with incomplete lineage sorting when constructing trees so that we're not fooled by incomplete lineage sorting. So we're going to build a unified model that looks at allele evolution in the context of locus evolution, in the context of gene duplication and loss within a species tree. And then lastly, we're going to look at how we can deal with recombination events and talk about ancestral recombination graphs, or ARGs. And then lastly, I'll touch upon a few points on the recent, over the last century, evolution of evolutionary theory and this whole modern synthesis, or the second modern synthesis more recently. So let's dive right in. First of all, how do we map gene trees to species trees? So basically, the way that you have to think about gene trees is that they're evolving inside a species tree. And in particular, if you look at everything that we've been studying so far in phylogenetics, we've always had branches unlabeled. A branch was a branch regardless of what species it came from. Right now, we're actually going to look at the fact that this is not just some random sequence, it's a gene from dog. And that's not some random sequence, it's a gene from human. So we're going to look at the species membership of the sequences that we're going to be aligning in order to understand how they're evolving inside the species trees. And in particular, if we had 2 different trees in the previous lecture that were showing, for example, a mouse and a rat gene in 2 copies, and this was in fact rooted all the way up here, then we would have had to infer that there was a loss event in the human lineage and a duplication event in the human mouse rat lineage because of the particular topology of the tree. And that's what reconciliation comes in. Basically, the species trees help us interpret the gene trees. Namely, if that branch roots, here or here, it's a very different kind of scenario, inferring different numbers of duplications and losses. So the main tools that we're going to be using is looking at the coalescence. Basically, we're looking at the common ancestor of any pair of sequences that we're comparing. And if 2 genes are finding their most recent common ancestor into a speciation event, then we're going to be talking about them as orthologs. If they find their most recent common ancestor in a duplication event, we're going to be talking about them as paralogs. So let's go through some definitions. So basically, the blue shade in the background is going to be the species tree. Every species is a bag of genes, and these genes evolve inside their bag. They don't duplicate outside the species context in which they live. And therefore, if there's a duplication event along a particular branch, it basically implies that in that lineage, the common ancestor of mouse and rat underwent duplication. And therefore, that every descendant along the mouse lineage or the rat lineage will basically have 2 copies unless there's a subsequent gene loss in one of those 2 copies. Therefore, duplication events affect all of the lineages that are crossing through there. Similarly, speciation events are speciating every single gene that is in that bag of genes, in that species. So every time I have a speciation between, say, the dog lineage or the human mouse and rat lineage, all of the genes speciate. You can't just choose not to speciate when the species separate. Is everybody with me about how the context in which we're studying these trees is in fact impacting the interpretation for both duplication events, which have to affect all of the lineages subsequent to that duplication event, and also speciation events that have to affect all of the genes at that speciation event? All right, so we're at 56.44.000. So we can therefore infer, based on these duplication events in star, speciation events in circles, we can infer that the black gene tree topology, when weaved into the blue species tree topology, implies a duplication event in the common ancestor of mouse and rat, a loss event in the human lineage, and then a speciation event in both mouse and rat. So that basically means that if you look at orthologs versus paralogs, we can actually start inferring which genes are functionally equivalent between species, or at least evolutionarily equivalent, and which genes are simply duplicates of an ancestral function. So why do we care about duplication loss? Because if 2 genes are related by a speciation event, we can kind of infer that they most likely play the same function in the 2 species, at least if the species are closely enough related to each other. So speciation events, and specifically orthologs, which basically is coming from the Greek orthos, which means straight, i.e. correct. So orthologs and paralogs means side by side, and then homologs simply means that they have homo means similar, para means side by side. Homologs basically are things that are just similar without distinguishing whether something is an ortholog or a paralog. So an ortholog is a gene that is most likely inferred to play the same function in the different species. And a paralog is a gene that is most likely inferred to have gained either gene dosage effects or potentially new functions for either one or both copies. So the reason why I'm a little ambiguous is that to truly prove orthology, you would need to do a replacement experiment. You would need to show that indeed these genes can functionally complement for each other if taken from one species and put into another species. And this is rarely going to be done. But instead, we're going to be talking about the evolutionary properties of genes that are related in a way that makes us infer that they most likely play the same function. So who's with me here on the definition of orthologs and paralogs and how that impacts our interpretation of the functional preservation of the function of those genes? So again, orthologs preserve the same function. Paralogs likely have the potential for gaining new functions. And orthologs are related by speciation events, whereas paralogs are related by duplication events. Okay, so 75, 25, 0, 0, 0. So now we can basically start thinking about what do these duplication events do. So the duplication events are in fact the fodder for new functions in the genome. So there's many things that can go on after a duplication event. So you could basically have neo-functionalization, which is that the second copy gains some new kind of function based on beneficial mutations that now allow it to do something else. Or both the ancestral copy and the recent copy might actually go undergo mutations that make them both gain either sub-functions or new functions, which are both different from the ancestral function. But most of the time, what you're going to have is after duplication, you're going to have non-functionalization, which basically means that one of the two copies will most likely be lost. And in the context of the whole genome duplication of yeast, as we talked about last, 2 lectures ago, we basically saw that the vast majority of genes of duplicated genes from the whole genome duplication had in fact been lost. That 90% of the duplicated genes were lost. So that's the most likely scenario. And then we saw that, a small subset of genes had actually subfunctionalization, and a much larger subset had shown neo-functionalization. So this is basically the building blocks for evolution, because now these two copy genes, if they're not lost, will most likely give rise to new functions. Now, does it make a difference when something is copy 1 or copy 2? Well, the answer is that copy 1, if it sits in the same location that it was initially in the genome, is most likely to preserve its gene regulatory circuitry. Whereas copy 2, if it's copied into another location in the genome, is most likely to lose the regulatory context. And therefore, copy 2, which will now be expressed in some other context, is much more likely to be the one undergoing neo-functionalization, whereas copy 1 is much more likely to preserve the ancestral function. But conversely, you can actually think about evolution of complexity as being relieved by duplication. So you can think of increased multifunctionality, increased pleiotropy of individual genes, which are then relieved after duplication, because you don't have to sort of be both, a coffee maker and a teapot in the same way. Basically, after you duplicate, one copy can specialize to become a teapot, and the other one can specialize to become a coffee maker. So whereas initially you may have just only been making coffee, for example, and now you have acquired this additional ability to make tea, but after duplication, you can now relieve these into the two subfunctions. So how often do gene duplications and losses occur? So we can actually estimate the rate of duplication and loss based on the age of paralogs via silent mutations. And Lynch and Connery basically did that, and also based on gene counts within families, and Hahn did that. And they were found to be extremely frequent and to be a major component of genome evolution. So duplications and losses, in fact, occurred in 10 to 40% of the rate of substitutions. So that means that it's enormous. You basically have new genes, at only 1/10 the rate of substitutions, which is, still quite remarkable. And there's a lot of variation between species. So duplications are faster in yeast with, 2 events every 1,000 years. which again is enormous. And it's a similar rate between mammals and flies, with roughly one event every 1,000 years. And you basically have a recent acceleration of these duplications in primates with 2 events every 1,000 years. And that's nearly 2-fold higher than other mammals, suggesting that there have been some unique evolutionary pressures acting in the primate lineage. If you look at what are some of the effects of duplication and loss, so basically neofunctionalization has given rise to tricolor vision. After duplication of one of the photoreceptors that was tuned to only one of the different colors, we now have tricolor vision in humans with a photoreceptor that is more tuned to the electromagnetic frequency corresponding to blue waves or green waves or red waves. And that's how we get our tricolor vision, basically based on the gain of one of the opsin genes through duplication. Similarly, there's been an expansion of olfactory receptors, which are smell receptors in rodents, and a contraction of those in primates. Primates, being able to see color much better, are able to recognize rotting fruit and recognize things that they don't need to eat, that they should not be eating, using vision rather than relying solely on smell. By contrast, rodents that are primarily nocturnal are able to smell extremely well with a huge expansion of olfactory receptors, which were in fact being lost in parallel along the human lineage. We have a lot of olfactory genes that are now pseudogenes that have lost their function. And again, last time or two times ago, we saw this extreme case of whole genome duplication about 150 million years ago in yeast. And these 2R hypothesis of 2 rounds of whole genome duplication near the core dates, giving rise to vertebrates and all of us, and also this whole genome duplication in teleos fish that we were part of showing. So here are some examples of gene families that are expanding, diverging. So you can see here, for example, these interleukin genes involved in immune response are in fact, expanding dramatically. So you basically have entire families that are duplicated and evolving and reduplicating within each of these lineages. And then similarly, these LDH genes are, very rapidly evolving. With the olfactory families, you see this dramatic expansion across multiple lineages. Again, this is one of the most common adaptations to new types of food sources. So flies adapt to different types of plants and fruits. Humans, rely on their vision, whereas all these other animals are relying on their olfaction to be able to recognize all these different types of food source. And then the chemical receptors are basically under adaptation to the new environment that each of these species inhabit. And then you can count here just how many gains and losses there have been along each of these lineages. And you can see here this dramatic number of gains and losses along each of those. So again, this is a golden era of comparative genomics. There's a large number of vertebrates, fish, amphibian, birds, mammals that are being sequenced. And it's an enormous opportunity for actually recognizing all of these events and how they have given rise to this incredible diversity of adaptation of different species in the planet. So gene duplication and loss are extremely important, and then inferring orthologs and paralogs is also extremely important. So how do we do that in more detail? The mechanism through which we do that is in fact reconciliation. The problem of reconciliation is how do we take a gene tree, which we know evolved inside a species tree, and actually weave the branches of the gene tree into the branches of the species tree to infer gene loss and gene duplication events. So the simplest algorithm for doing that is actually maximum parsimony reconciliation. So reconciliation is basically taking a gene tree and a species tree and reconciling them with each other, basically mapping one onto the other. And the problem of reconciliation is mapping recursively the reconciliation node of every node in the gene tree. So every species maps into itself. Okay, so if it's a rat, only it maps into rat. And then where it gets interesting is that the reconciliation of a particular node is the least common ancestor of the reconciliation of the right node and the reconciliation of the left node. Okay, so if mouse maps to mouse and rat maps to rat, then the reconciliation of that node, which is an internal node, will basically be mapping to the least common ancestor to the most recent common ancestor of the two species. Okay, so we're going to be basically so if it's a leaf, then it simply maps to species, and if it's an internal node, it simply maps to the common ancestor of the two. Okay, so that basically means that if I can basically start reconciling from the bottom up, moving up the tree, and then moving up the reconciliation events. So this node here reconciles there, and this node here also reconciles there, and this node here also reconciles there. So basically all these three internal nodes are in fact reconciling to the same spot of that tree. And now we can label these events as duplications if the reconciliation of a node V is in fact the same as either the reconciliation of the left or the right. So that basically means that this event here, this star that gets mapped to that same location, is in fact a duplication event because it has the same reconciliation as either of its children, and in this particular example, as both of its children. And if the branch above V has at least one loss, then, the branch above V has at least one loss if the parent of V does not reconcile to the same location as V itself. So we can infer a loss here if the parent of this one doesn't reconcile to the same location as that one. So basically you can see here that there's in fact one missing reconciliation. And because after reconciling this node here, this duplication node here, we basically have to jump over a particular species, that basically means that there was a loss along that lineage. Okay. So who's with me here on this very simple algorithm of reconciliation, basically reconciling every node to the species that it comes from, and then moving up the tree to reconcile additional events as duplication events if the left or the right matches, or loss events if the parent is in fact absent and you're skipping one between them. So we're at 44, 56, 0, 0, 0. Okay. So how do we now infer events inside a gene family? So basically what we need to do is start understanding how we have, for example, a set of sequences with a dog sequence, a mouse sequence, a rat sequence, another mouse sequence, and another rat sequence. We want to now align all of those, infer a gene tree, and then map the gene tree by reconciliation. So if we're trying to understand the evolution of a gene family, we basically first reconstruct the tree and then reconcile that tree to the species tree. Of course, as we're going to see later in the lecture, reconciling these can lead to errors because of all kinds of reasons, and therefore doing it in two steps might be the wrong choice. And we're going to look at some phylogenomic methods for doing the two steps jointly. So again, the reconciliation problem, or the maximum reconciliation problem, is given a gene tree G and a species tree S, find the reconciliation that implies the fewest duplication and or loss events. And there's many algorithms for finding these reconciliations. The first one was proposed by Goodman. Parsimony was proposed in 1994 with minimizing duplications and loss. A linear time version of that was proposed a few years later. And then there was a proof of the minimum and the uniqueness a few years after that. And then Bayesian methods for reconciliation were also proposed in parallel. So here's some examples to gain intuition about reconciliation. So if you have a one-to-one mapping between these tree and that species tree, then there's zero duplications, zero losses, and every pair of genes is in fact orthologous. But in fact, there's, if I swap, for example, the D and the H branches, so all I'm doing here is swapping two branches in the gene tree. And this kind of swapping could happen simply because of a mistake. Perhaps this branch is very short, and in that location, human had a few more mutations that pushed it up the tree, and therefore human was rooted outside. But what does that mean for reconciliation? That basically means that suddenly, because of this common ancestor of dog, mouse, rat, and human basically mapping here, and this ancestor mapping here, that basically means that there was an ancestral duplication event, followed by speciation, followed by loss in dog, loss in human, and loss in the mouse rat ancestor. So basically, if we make the slightest mistake in the gene tree, we can result into 1 duplication and 3 losses and a lot of sort of paralogous relationships simply because of that. So that's why it's extremely important to really understand the confidence with which we can say that there was, in fact, a different topology. Does everybody see this enormous impact that just one swapping can have on the numbers of inferred duplications and losses based on this reconciliation approach? Okay, so we're at 2278,000. Okay, if there's any questions, I'm happy to take them now. There's no questions. Okay, so if it's a non-parsimonious reconciliation, for example, if now human is up there, if it's incorrectly reconciled, then I can infer multiple duplications and losses. But in fact, this reconciliation is not minimum cost. So basically, we're talking about the reconciliation R that implies the fewest duplications and loss events. So even if dog, human, mouse, rat are in fact congruous between the two, if I incorrectly reconcile the human branch up here, then that basically means that I will infer 2 duplications or 4 loss events, even though my gene tree is correct. So that's why it's important to have a correct reconciliation. Again, valid reconciliations must respect the descendant-ancestor relationship. So every time I map an event, it better be in the same order if I go from the leaves to the root, rather than crossing any of the previously mapped events. All right, so I highlighted the importance of building correct gene trees in inferring duplications and losses. What we would love to do now is see how we can leverage the genome-wide nature of these trees to basically get into phylogenomics. And again, omics means the entire genome at a time. So phylogenomics means phylogeny, but at the level of entire genomes. So let's look at gene tree and species tree reconstruction in the context of phylogenomics. So the first challenge is, how do we build an accurate species tree? And one way to build a species tree is to just take one random gene in the genome, build a tree for that one random gene, and say, great, the hemoglobin gene gives me that species tree, and therefore I'm done. But the challenge, of course, is that any one gene tree might actually have small discrepancies. And therefore, we might actually want to leverage multiple locations of the genome to gain a more accurate view of what is the most likely species tree relating the species that we're looking at. So the goal is, given a set of gene trees, find a species tree that either is a consensus of the gene trees or is built from concatenating all of the gene alignments, or has the minimum number of implied duplications and losses onto the gene trees, or the minimum number of deep coalescences, etc. And the key insight is that we're going to be wanting to combine information from the entire genome to build a species tree, and therefore we must decide how to combine conflicting information. So again, we can align any one gene family at a time and then build a gene tree for those, and then start asking how many splits are there between every branch and every internal branch. So basically, if you look here, the internal branch 1 is in fact a dichotomy between species A and B on one side and species C and D on the other side. And even if E and F are not implicated, we can basically say that it gives us a vector where half the species are on one side and half the species are on the other side. Similarly, branch 2 and branch 3 here can distinguish. Branch 2 distinguishes AB on one side from CDE on the other side. And then branch 3 distinguishes ABC on one side and DE on the other side. Branch 4 here distinguishes AC from DF, branch 4 AC from DF, and branch 5 CD from EF. Okay, so with any one of those, what you can see is that they don't necessarily agree with each other. You can't just simply put them all together. But what you can do is ask how many splits are in fact supported by each of these trees, and therefore start inferring a supertree that best captures the observed splits, these internal branches. And then the supertree is shown here, where even though there isn't a single tree that captures all of the species, we can still infer a supertree that captures all of the splits. So who's with me so far on the concept that every branch is a split between a subset of species, that we can represent all of this in this matrix called the Baum-Ragan matrix, and that we can use this matrix to infer a supertree that best captures all of these splits. So again, every bifurcation event can be represented, even if they're conflicting with each other. You can just simply say, well, how many gene trees were in fact supporting the 1 split or the 2 split or the 3 split or the 4 split or the 5 split, and so on and so forth. And based on the number of trees that support each of these splits, you can in the end count what are the most supported splits, and then place these splits into a tree. So we're at 25, 50, 13, 13, 0. Okay, so what are some methods for doing these species tree reconstructions? So there are super matrix methods, and these super matrix methods are basically asking, if I have matrices of similarities across each of these trees and across each of the species, can I have a combined score for the overall distance between any pair of species through that matrix? Another one is a super tree method, which is basically looking at the splits and then the trees that sort of best capture the splits that are most frequently found. Another way is to minimize deep coalescence, which we're going to talk about. Basically, how do you minimize the number of events, and also to explicitly model coalescence. And another one is to simply concatenate the gene alignment. So I can basically take, instead of having one tree here and one tree here and one tree here and one tree here, I could just simply concatenate all the alignments and just build a single tree, perhaps with some bootstrapping to basically understand the variability of that tree from that, or build minimum number of implied duplications and losses. So what does that mean? That basically means that I can choose a species tree that seems correct, maybe from this method, and then evaluate what are the total number of duplications and losses that we infer when reconciling each of the gene trees into that species tree. And after I've inferred all of these reconciliations, I can have a total cost for all of the gene trees, and then infer whether that species tree has a minimum cost. And then I can take some additional lower ranked species trees and then see if they have the minimum number of implied duplications and losses from that. Okay. So who's with me here on the species tree inference using combinations of many, many different gene trees? So we can use either concatenation or a combined distance matrix or a minimum number of events according to coalescence, duplication loss, and so on and so forth. Okay, so 13, 40, 50, 00. So that's at the species tree level. We have now gathered all of this information across many, many different gene trees or gene alignments to infer a species tree. And now we can basically say, well, can I infer a gene tree more accurately using the species tree? So the challenge here is that we want to use the species tree to actually improve gene tree reconstruction. We can't just simply say, oh, let's just build a gene tree and then reconcile it and then we're done. What we want is algorithms that no longer treat sequences as anonymous, ignoring what species they came from, as we saw earlier. Instead, if we know the species, we can define some prior distributions of reasonable gene topologies and branch lengths. So that's the key insight that I want you guys to have. If I have built 300 gene trees already, when I build my 301st gene tree, I can leverage the information from the previous 300 to basically say, well, I do expect the species to branch in this order. And I do expect the lengths of each of these branches to be roughly this proportion of each other, and so on and so forth. So what we're going to do is develop an evolutionary rate model that will basically predict the probability of observing a particular set of divergence times and rates for each of the species, given the topology, the reconciliation, the species tree, and a set of parameters. So if I basically have, 5,000 gene trees across the genome that are all involving the same set of species, then I have no reason to expect that every single gene has evolved according to its own history, because these bags of genes evolved together in the same set of species. So we can basically use that to study the properties of gene trees at different locations. And what you see is that if you study this particular gene here, you see very long branches throughout the tree. If you study this gene here, you basically see very short branches throughout the tree. So that basically suggests that perhaps there's a normalization possibility of normalizing all of the trees together, and a scaling factor through which this normalized tree would be scaled with either a slow evolving gene or a fast evolving gene. Who's with me here on the concept that these gene trees are all sampled from the same underlying distribution of gene trees from that set of species? And as I do that sampling, I can basically scale them according to some genes that are simply evolving faster, perhaps because they're more involved in environmental adaptation, versus genes that are evolving slower, perhaps because they are, involved with histone function or something like that, which generally evolves very slowly, regardless of the species that they're in. So we're at 25, 63, 13, 00. So that basically says that we can actually start modeling the process through which these gene trees evolve inside species trees. So what we're going to be developing is this rates model of an independent species-specific rate, which is scaled by a gene-specific rate. So we can basically look at the absolute lengths of any pair of species. For example, Drosophila ananasi and Drosophila grimshawi, which are quite far away in this tree, and basically ask across 5,000 trees, what is the length of the ananasi branch and what is the length of the grimshawi branch? And even though these species are very, very far from each other, the correlation between them is actually 0.68. So that basically means that those genes that evolve faster in that species are also evolving faster in that species. And this is just the average correlation between any pair of species, any pair of branches, even internal branches denoted here. And what you can see is that the average correlation is in fact 0.56. That suggests that perhaps we should be scaling these branches across the entire tree, basically take every single tree and scale it to have the same total number of relative evolution after extracting out the fact that some genes evolve faster and some genes evolve slower. And that basically indeed shows this capturing of this independence between these two branches, which are now only 0.08 correlation. And indeed, if you look at this entire tree, the average correlation is now only 0.09. So who's with me so far on what we've done? We've basically now normalized all of the trees by the total number of substitutions per site at that tree. And that gives us a relative rate of divergence. which basically is now much more independent between pairs of species. So we're at 1767, 1700. All right, so that basically gives us this rates model, which has an inverse gamma distribution for the gene rate. So some genes evolve faster, some genes evolve slower. And if you look at the scaling factor and how it is distributed, you see this black curve of the actual empirically measured scalings. And it kind of fits very nicely with this either gamma distribution in blue or a little more closely with this inverse gamma distribution in red. So that's the gene rate. And after I've corrected for the gene rate, the resulting rates of these species residuals are in fact gamma distributed. And you can basically see here these gamma distributions that are matching quite well. You see this green curve is the gamma distribution, and then the red histogram is how well the distribution of relative lengths fits along each of these branches. So that's kind of cool. We can basically express the length of any branch as this rate times the divergence time, which is basically a gene rate times the species rate times the divergence time. So basically there's been so much time elapsed along this Grimshaw-Wheel lineage, but that time elapsed, TI, is in fact scaled by the species-specific evolutionary rate and the gene-specific evolutionary rate. So basically to infer the rate of evolution of any one branch, I want to multiply the gene rate, the species rate, and the time that the branch captures. Okay, so let's see who's with me so far. Lovely. So 0, 71, 30, 0, 0. Okay, so the traditional phylogenomic pipeline has been to take all of the gene sequences, blast everything against everything using lecture 3, then cluster everything into gene clusters using our clustering lecture, and then construct alignments for each cluster using our lecture 2, and then build trees using last time's lecture, and then reconcile using the beginning of today's lecture. But the species tree only comes at the end when it's time for reconciliation. And then based on that reconciliation, you can infer reconciled trees, orthologs, paralogs, duplications, and losses. And that's how most methods are actually working. And there's a lot of databases and methods for doing this, and they're all using the species tree only at the end. But what we can do now is actually use the species tree earlier. What we can do is actually guide the types of trees that we would expect using the species tree, and basically have a generative model that is informed by the species tree, and then infer the Bayesian probability of a gene tree using the prior for what I would expect given the species tree, and the evidence from the particular sequence that we're looking at to calculate the likelihood and the posterior probability of that species, of that gene tree for that set of species. So what we're going to be building is basically this species-informed maximum a posteriori method, or SPI-MAP. Okay, and this is work that Rasmussen and myself developed in molecular biology and evolution in 2010. So basically, given a species tree, we can basically build this generative model that has a duplication and loss model that infers times of divergence and times of duplication and times of loss given this generative model. And again, the goal is not to simulate, but to infer the posterior probability of having observed that given ultimately the data. But having a generative model allows us to now use Bayesian inference to infer the most likely tree overall using the probability that indeed the particular topology that I'm proposing could have been generated from that species tree and this evolutionary model. So that's the first step, having a duplication and loss model. The second step is basically having now a rates model that is the product of a gene tree specific rate and the divergence times. And then this rate here is both the gene tree and the species specific rates. So we basically now have a rates model that generates rates of evolution. And then your sequence specific model, and this could be general as HKY or Juice-Cantor or Kimura two-parameter model and so on and so forth, which basically generates sequences for each of the leaves that match the current sequence. So given this forward generative model, I can then ask, okay, given that I have a set of sequences, what is the posterior probability of a particular set of, rates, trees, topologies, duplications, losses, etc. And if I propose multiple different types of models, I can then score the posterior probability of each of them given the sequence alignment data information. So who's with me so far on this complete model that's sort of putting all these pieces together, the species tree, the duplication and loss model, the rates model, and ultimately the sequence model. So all of that in a generative framework that allows us to now propose different types of topologies, lengths, rates, times of divergence, and then evaluate them in the context of the observed data using Bayesian inference and maximum posteriority estimation. So we're at 0, 70, 30, 0, 0. Okay. So here's how the reconstruction actually works using this posterior probability model. So we want to find the maximum a posteriori tree, which is basically a set of branch lengths, L, a set of a tree topology, T, reconciliation of how that tree topology maps onto the species tree, which is the argmax searching over these three options, given the data and a set of species tree and other model parameters. Okay, so that posterior probability is nothing but the probability of generating the data given the branch length and the tree topology. And that's basically given our sequence model and this pruning algorithm. And the prior for both the lengths and the topology. And then what is the branch length prior? That branch length prior can be numerically computed based on simply what is the probability of sampling, say, a length which is this long from that distribution. So I have the distribution, I observe the length, and I say, well, that length would have been sampled according to that probability. And similarly, if I have a distribution of gene rate, I can infer where, this is what I observed, this is the probability with which I would have generated. Okay, so that is numerically computed. And then the topology prior is based on the duplication loss model, and that's computed using a dynamic programming algorithm. Okay, so now we can now start turning into how do we compute all these models? How do we, we basically have all of the components for this based on the phylogenetics lecture from before. This is nothing but this dynamic programming walking up the tree, where we're basically saying what is the probability of observing a set of sequence alignments given a set of lengths. And what we saw in the phylogenetics lecture is how we can in fact treat every column as independent. What we can do is marginalize over all of the ancestral species settings and then start asking for every setting of ACGT at every point as I go up the tree, what is the cost given this probabilistic model, this log score that I'm adding up the tree to basically infer what would be the cost for each of the settings of A, C, G, and T. And then I basically have a forward pass, which is this assignment of each of these ancestral nodes or a computation that marginalizes over these ancestral nodes. So all of that we already have from our previous lecture. And then we already have this using our branch prior that we just talked about. And then we already have that from this duplication loss model. So we can compute all this using this maximum posteriority method. So let's see how does this method perform. So if you look at, this neighbor joining algorithm that we saw last time, it basically infers only 60% of orthologs correctly, and it infers 22,000 duplications and 71,000 losses, given whole genome alignment of all of these trees. So that's what we need to surpass. And now this maximum likelihood approach, or this posterior probability approach, that are not based on the species tree, are doing slightly better and inferring slightly fewer duplications and losses compared to that. But again, they're taking a lot longer, so about 80 times longer. And now we can basically see how are these species-informed methods doing. So this spider method, which is this maximum likelihood method, is already improving from 60% or 63% to 83%, and inferring only half of the duplication events and only half of the loss events, suggesting that in fact it is able to explain the alignments with many fewer events and therefore much more accurately. And then this SPI-MAP method, this maximum a posteriori species-informed method, using different kind of settings, or a quick search or a long search, is in fact inferring a fourfold reduction in the number of duplications and the number of losses, and a dramatically higher number of orthologs and paralogs. So it makes a very big difference to be able to incorporate the species tree in being able to infer the evolutionary scenario of every gene in the genome. All right, so who's with me here? So what we've basically done is that we've used the species tree to guide the scoring function for any kind of gene tree topology that I'm proposing, instead of only at the end using the species tree to say, hey, did we reconcile it correctly or not? But now we're using it from the beginning in the context of this generative model of sampling sequences from these rates model, these times model, and the species tree model. All right, so we're at 0, 70, 30, 0, 0. Okay, so this is basically looking at number one, reconciliation, this whole concept of having gene trees and species trees as separate types of objects. And then the concept of learning across the entire genome to infer, number one, a high quality species tree by combining multiple gene trees and multiple gene alignments simultaneously, and also higher confidence gene trees using this scaling factor to basically tell us how are genes distributed in their evolutionary rate, and also learning the species-specific distributions and the gene-specific distributions in the context of this generative model. And we saw this dramatically improved accuracy for gene tree reconstruction in the context of species trees. Let's now switch gears and look at evolution in the context of populations. Again, the evolutionary process that we've talked about are not simply between a single individual that then decides to become a rat or a dog and so on and so forth, but it's a population of individuals that are evolving. So what we need to do is adapt phylogenetic methods for shorter timescales to deal with population effects. And there are many different models for populations. We're going to look at a time forward model of the Wright-Fisher model, and that's used to study the effect of drift, for example, and also a time running backwards model, which is the coalescent model, and that's a much more efficient approximation for working with trees of individuals. So let's start with the Wright-Fisher model. The goal of the Wright-Fisher model is to model the stochastic behavior of population structure, and it was developed by Wright, about 90 years ago. And it was used to study the effect of drift on allele frequency and migration, population bottlenecks, selection, and also the age of the most recent common ancestor. So then it makes a number of assumptions. It basically assumes that there's a fixed population size n, that there's random mating, that it's all neutral, there's no selection, either positive or negative, and that there are non-overlapping generations. And the process is that every individual generates an infinite number of gametes, and we randomly choose gametes to make n individuals in the next generation. And an equivalent process would be that every chromosome is chosen randomly with replacement from the previous generation. So here's every individual with their 2 chromosomes. And this individual contributed this chromosome to that individual and that chromosome to this individual. And that individual contributed this chromosome here. And that individual, for example, contributed 1 chromosome, but the other chromosome was just lost. It didn't make it to any of the descendants. And that's simply a product of meiosis. You can basically say, if I have these meiosis events, you saw how, I could I could inherit either my dad's paternal chromosome or my dad's maternal chromosome. If I only inherit my dad's paternal chromosome, my dad's maternal chromosome is lost from the gene pool. It's no longer to be found. And that's what this model is capturing. The fact that at every generation, I have some chance of inheriting, of passing on my, my DNA. So going forward in time, every one of those might or might not make it to the next generation. And any one of them can actually give rise to multiple progeny, okay? It just so happens that this 1 chromosome now made it to the next generation into 4 different chromosomes, okay? Who's with me so far on this model here? Okay, this is a forward-looking model that is basically asking what is the fate of every chromosome in a pool of individuals. So we're at 43, 43, 14, 0, 0. Now, if you notice backward in time, however, every individual has exactly 1 ancestor. So because every chromosome came from exactly 1 place, okay? So that's now looking at this model, which seems fairly complex if we use the boundaries separating every person. But you can sort of trace that across many generations, and then think of this as this very messy splitting of chromosomes across different generations. Going back in time, every one of them has exactly 1 ancestor. Going forward in time, many of them will be lost, and many others will have multiple descendants. But if we abolish the individual boundaries and we just think about chromosomes evolving, instead of having n individuals, I now have 2n chromosomes. And every chromosome basically gives rise to 1 chromosome. Going backward in time, every chromosome came from exactly 1 chromosome. And going forward in time, every chromosome can give rise to 0 or more chromosomes. But the beauty of having gotten rid of these little circles is that I can now sort the tree so that there are no crosses. And that suddenly makes it so much cleaner. So what that tells you is that this 1 chromosome gave rise to 3, this one was lost, this one came down all the way here, this one came down all the way there, and then some of them that made it all the way down, you can trace them all the way up. But if you take any random one of them, chances are that you're going to be losing it somewhere. So this is one method and model that has been used to basically say that, yes, even though there are 7 billion people on the planet, any one of us can trace their relatives only so far back in time before all of the chromosomes coalesce. And therefore that the true ancestor of all modern humans is in fact much, much more recent than these first modern humans. So what's really cool is that if you forward in time, you have a hard time finding lineages that make it all the way to the end. But if you trace back in time, you can actually have a much simpler way of modeling all of these processes. So we can randomly sample extant chromosomes, so chromosomes that are still around today, and then trace them back until they coalesce in the tree. And that's the coalescence model that we're going to be looking at. So the Wright-Fisher model is basically moving forward in time, but the coalescence model is moving backward in time. What we can do now with this coalescence model is very cool. We can actually ask, after how many generations are 2 random chromosomes coalescing into an ancestral chromosome, and so on and so forth. So before we move on, who's with me here on this Wright-Fisher model moving forward? This very complicated model with n individuals each being deployed and the chromosomes intercrossing like crazy, and this much simpler model of 2n chromosomes, and then allowing to reshuffle so that there's no crossings, and then tracing down going forward in time with Wright-Fisher or going backward in time with coalescent, the ancestry of every extant lineage. So we're at 33, 50, 17, 00. All right, so we can now start using this coalescent model to ask what are the time distributions of these coalescent events and how do we simulate coalescent events going backward in time. So we only need to now model the genealogy. We can think about time going backwards and lineages coalescing. So basically the question is, when do I get the first coalescence event? When do I get the second coalescence event? And when do I get the third coalescence event? So the coalescent model basically only focuses on the distribution of coalescence times, Ti, going back in time. And every pair of lineages has equal probability of coalescence. And we can approximate for large N, every Ti will also be large. And we can treat the coalescence time as a continuous variable rather than a discrete number of generation events. So now we can ask, what is the probability that K lineages don't have any coalescent events in the parental generation? And that's basically the probability of not coalescing for the first one, not coalescing for the second one, not coalescing for the kth one, which is basically the product from 1 all the way to Kn minus 1 of 1 minus I over 2n. Okay, so that's simply, I have n chances of coalescing. I don't meet any one of them. That's basically the chance of not coalescing. Okay, and that can be basically factored out into 1 minus this plus some kind of factor which is order of 1 over n squared, and basically this is k choose 2, times 1 over 2n, basically by summing over this j. Okay, so for k much smaller than n, then this order of n squared is very small, and therefore the part that dominates is this one. So it's basically the probability of coalescing is given by the size of the population and then the number of lineages that we're looking to coalesce. So we can ask, what is the probability that the first coalescence of K lineages happens after T generations? And that's basically using that property that we saw before, depending on the number of lineages and the number of individuals in the population, and then you run that after T minus 1 generations. And then that's nothing more than the geometric function of K choose 2, 1 over 2n, which can be approximated by an exponential function. And therefore, for K lineages coalescing, which is much smaller than the number of individuals in the population, the probability of multiple coalescence in one generation is actually very small. And for large times, the geometric distribution well approximates this exponential. Okay, so basically, this is a geometric drop-off of how slowly things will eventually coalesce. And you can think of it as a memory-based process moving back in time at every generation, basically having the same probability of coalescing. So we can actually simulate this coalescent process by sort of asking, well, if there's 100 lineages or if there's 10 lineages, what are the distributions of coalescence events? And you can project that down to basically see with what rate are these coalescence events happening earlier or later for different numbers of lineages. So having now this tool for modeling coalescence time distributions and simulating coalescence, we can actually tackle head-on incomplete lineage sorting. Okay, so first we're going to define incomplete lineage sorting, and then we're going to use increasingly sophisticated methods for dealing with it. So the first model that we need to have is this multi-species coalescence. And again, the idea here is that every branch on this blue tree that we were looking at, the species tree, is in fact made out of individuals evolving in that population. And when we trace genes going back in time and their coalescence, these are traced through individuals in the population. And that basically means that if the population is extremely large, this probability of two lineages actually coalescing is quite small. If population has only been around for, I don't know, 6 generations, and there are, I don't know, 100 million individuals in that population, then the probability that any two alleles coalesce before we switch over to the next lineage in our tree is actually quite small. So the larger the number of individuals and the shorter the length of time that was passed in that species boundary, the lower the chance of actually coalescing. And that basically gives you a coalescent event here, a coalescent event there, but a deep coalescence event where this blue lineage and this red lineage don't coalesce in this population, but in fact they go further back in time and they don't coalesce until up here. But what's really interesting is that this red lineage, in fact, coalesces with a black lineage earlier than the blue coalescence with the black lineage. So that's a little counterintuitive. There's nothing wrong with our tree, but in fact, there's a deep coalescence event. And basically, when I build my gene tree with species, with examples from species A, species B, species C, species C, D, I end up with individual genes from A, B, C, and D coalescing in a tree topology that is incongruent with the species topology. So basically what we end up with is gene topology, which has A and B be sister lineages, C and D be sister lineages, and then the ancestor being apparently a split between AB and then CD on one side and the other side. So who's with me here? Who can see this deep coalescence event and sort of how the population genetics properties of each population are in fact dictating the probability of actually coalescing within the span of interval that we're looking at here. So we're at 20, 60, 20, 0, 0. So let's now model this deep coalescent model going through these incomplete lineage sorting events. Okay, so why do we call this incomplete lineage sorting? Because the lineages have not fully been sorted out in sort of going forward in time. So basically, the population here continues to maintain the ancestral diversity of the blue population into the green population. And that ancestral diversity has basically, been further speciated before that species became pan-mixed. So that's the forward view of incomplete lineage sorting. So basically, if you evolve things, they just don't sort out fully within that branch. Another way of thinking about incomplete lineage sorting is deep coalescence. And that basically means that as I'm looking backward in time, these don't coalesce within that species, but they coalesce further, more deeply in time. Okay, so incomplete lineage sorting and deep coalescence are basically the same thing, just a forward in time view versus a backward in time view. So looking for these types of discrepancies between gene trees and species trees, Mike Eisen and colleagues basically looked at these topologies for Drosophila and then inferred widespread discordance between gene trees and species trees, suggesting incomplete lineage sorting. So they basically said, well, that basically means that the population of Drosophila species has maintained its diversity across speciation boundaries, leading to these deep coalescence events. Similarly, in primates, this group has basically proposed that genomic relationships and speciation times of human, chimp, and gorilla infer a coalescent relationship, where you basically have some topologies of human, chimp, and gorilla being incongruent with the species topology. And that's simply because the ancestral population level variability between individuals of the same population has been maintained through these speciation events that happen relatively recently and in relatively short times. So given the population sizes of the human chimpanzees or a human chimp gorilla ancestor, around 50% of the human genome coalesces with chimpanzee after speciation with gorilla, given a generation time of 25 years and this amount of time between speciation. You can basically build this hidden Markov model that allows you to infer which topologies are more or less likely to occur. And then again, incomplete lineage sorting has been used to infer species trees, divergence times, and population sizes. So you can estimate species divergence times and ancestral population sizes using DNA sequences from multiple loci. You can infer high-resolution species trees using this approach that deals with concatenation, and then that does not have to resort to concatenation by using this sort of deep coalescence. And then you can also infer species trees by minimizing deep coalescence rather than minimizing duplication and loss events. And then that incomplete lineage sorting has also been used to determine the human population tree by inferring ancient human demography from individual genome sequences. So what you can do is actually estimate the divergence times of human populations and migration rates using a coalescent model with many small non-recombining neutral loci. So you can basically say how many loci coalesce at different times. And based on this coalescence probability, I can infer the population size at every time point in the past based on when any two genes coalesce. So that gives me these distributions of population sizes that I was showing you in previous lectures, where you basically have this very large population size and then this initial expansion followed by a bottleneck, followed by these large population-specific expansions in recent history. Okay, so that's using incomplete lineage sorting to infer species trees, divergence times, population sizes, and so on and so forth. But what we can also do is start building a unified model of allele evolution, locus evolution, and species evolution in the same context. Let's go back to our two views of gene evolution that we saw so far in this lecture. One of them was duplication and loss, where incongruence between a gene tree and a species tree was interpreted as a duplication event followed by loss, loss, and loss. Another view, which we saw in the third section of the lecture, was coalescence, where the incongruence between the ABC topology of the gene tree that does not match the species tree is simply due to these two genes that are found in this lineage and that lineage simply crossing through this species boundary before coalescing after B coalesces with A. So if we have an incongruent gene tree and species tree, there's really two orthogonal ways of interpreting this. One is duplication and loss, the other one is deep coalescence. And there are simply no models that allow you to model duplication and loss jointly along with coalescence. The coalescent models basically deal with one-to-one orthologs, and they estimate divergence times, population sizes, etc. And these models move backward in time with coalescence, and they cannot cope with duplication and loss. If you just duplicate a gene, there's just nothing you can do to model it. But duplication and loss models, in contrast, deal with paralogous families, and they estimate birth and death rates. And these models move forward in time, and they can simply not cope with incomplete lineage sorting. They just simply break down. They have no modeling power for resolving this. So again, what Matt Rasmussen and I did was build this DL-Cole model that actually blends together duplication and loss and deep coalescence into a single unified framework. The idea was very simple. So you basically have a locus tree evolving inside a species tree. So duplication events create new loci and new alleles, and this new locus can then go on to fixation, extinction, or remain polymorphic in the population. So you basically have a duplication event that happens, and that duplication event affects one individual where that happens. that one individual will then have progeny, shown in black, which might be fully penetrant, basically, take over that population, or be simply lost because of this random mating and segregation of chromosomes and alleles in the population. So you can basically have this duplication evolving inside the context of a population. So the original locus can go extinct, remain fixed, or become polymorphic, or remain polymorphic. So basically, after every duplication event, you have two fates for each of the subsequent lineages. Who's with me here on this concept of a locus tree evolving inside a species tree, and then these alleles inside the locus tree being able to be traced back in time in the same way that we would do for traditional coalescent model, but now with the ability to model duplications and losses. So we're at 20, 50, 30, 0, 0. So but what we basically need to do now is have a coalescent tree, which moves back in time, coalesce inside a locus tree, which moves forward in time, which coalesces inside a species tree. So we're going to have a duplication loss model in this way, moving forward in time, and a coalescent model between them two, moving backward in time. So that allows us to now model all kinds of events between a particular locus duplicating and then remaining, or being lost in one of them, or being lost in the other, and so on and so forth. So that's basically dealing with this unified model of duplication, loss, and coalescence, and enabling us to have trees that move both forward and backward in time simultaneously. The next question is, how do we deal now with recombination events? So what are these recombination events? So remember the tragic love story of PRDM9 that's basically guiding the search for its motif, its beloved motif in the genome. And every time it finds that motif, it cuts it, and it leads to repair of that motif from the other allele, and thereby loss of its beloved motif and its favorite motif. And therefore, this very rapid evolution of the protein, but also the resolving of these cutting of the DNA by repair, and half the time crossover, where this chromosome will now continue with that chromosome. So what does that mean? That basically means that the recombination events that are happening between pairs of chromosomes are causing the history to no longer be tree-like. In a tree, every node has one parent, except the root. But with recombination, a sequence has actually two parents, one from here, one from there. So the way that we can think about this is that in this part of the tree, the history is one way, and in that part of the tree, the history is another way. So at every recombination event, the topology of which parent I chose at any one location can actually shift. So that basically means that as I'm scanning along the genome at nucleotide 1 or at gene 1, I might have one topology. And as I'm moving along the genome, I'm basically shifting to a recombination event that now makes the paternal, the ancestral, the parental ancestor of C be connected to another location. And then as I move further up, this tree can, sort of yet go through another location and so forth. So what we can basically think about is this process of coalescence moving sequentially from left to right on the genome and modeled according to each of these recombination events. So every recombination event basically leads to a change in the topology. And we can basically use this to infer the specific set of recombinations that occurred between any set of individuals. So you can now use a hidden Markov model to move along the genome, knowing the state that you're in of which recombination tree you're using at every location, and then shifting from one to the other as you go from location to location, with the specific recombination event being the transition between one tree and the other tree. So you basically have a state transition probability based on the cost of that recombination event, a hidden state, which basically tells you the topology of every place along the genome, and then this sequentially Markovian nature of switching with the same probability at every location in a memoryless fashion. So you can basically now use this. So first of all, who's with me here on this ancestral recombination graph model that is moving from left to right on the genome, inferring the topology of every location, and then switching topology to topology based on transitions between these ancestral states that correspond to topologies based on these recombination events that basically regraft the tree from one branch to another branch. So we're at 0, 80, 0, 20, 0. Okay, so we can now model coalescence and recombination processes. We can basically use the sequential approximation of this sequentially Markov coalescent or col-HMM, or this conditional sampling distribution and pairwise sequentially Markovian coalescent models that are basically asking, as I move from left to right, what are the most likely interpretations for the topology at any one of the states? And you can basically infer genealogies moving along the chromosome by switching from one topology to the other. And then here you can see that all of these genes are being the blue topology, then the red topology, and so on and so forth, based on individual recombination events. So this basically allows you to now construct these ancestral recombination graphs that are telling you about every recombination event across the entire topology of all of the individuals that are linked together. So in summary, we basically looked at the reconciliation problem of basically mapping a gene tree to a species tree, about species tree reconstruction given a set of gene trees, and about gene tree reconstruction given a species tree. We looked at phylogenomics for building species trees more robustly and also building gene trees more robustly if we know the relationship between gene trees and species trees. And then models of population level evolution using the Wright-Fisher model that moves forward in time, the coalescent model that moves backward in time, and this multi-species coalescent that starts combining the two. And then this unified model of alleles evolving inside loci, evolving inside species, this mapping backward in time, and this mapping forward in time with duplications and losses. And we talked a little bit about sort of the evolution of evolutionary thinking across the centuries and how that led to modern genetics. But what we haven't talked about is this continued progression with increasingly complex models of evolution that are incorporating, all of these additionally complex models of evolutionary theory. So I'll leave this slide in the lecture notes so that you can look up individual models for your own interest. Of course, this is not going to be covered in the quiz, but this is something that for your own interest is worth looking at, sort of the evolution of evolutionary theory across, the effect of the adaptive landscape, of evo-devo, of recessive genes, and this reservoir of genetic variability, this relationship between pollution and the colors of moths, and the realization that evolution is in fact very fast-acting and testable and quantifiable. We already talked about Fisher and this unification of these continuous traits that are simply governed by many Mendelian loci, and therefore the Darwinian selection can still act within that. Morgan before that was basically looking at fruit flies and heredity and very small Mendelian factors still supporting Darwin. And Castle was looking at this back crossing, which in fact strengthens the initial rat strains with a larger hood. So that basically means that this discrete evolution and discrete inheritance that Mendel had in fact proposed was probably right. And this concept of the germplasm, of distinguishing germline versus somatic, that in fact the germline does not get to interact with the environment except through selection, through the soma that it produces. And, of course, Darwin with natural selection, and then Mendel with dominant recessive and also independent assortment. Wright basically talked about this adaptive landscape and how we can basically move into local maxima and local minima. And how drift can in fact take you from one local optimum to another local optimum that perhaps might be inferior through neutral evolution. Dobzhansky was basically looking at climate effects on evolution, and then the relationship between cell biology, drift, migration, isolation, and sterility. Ford was looking at ecological genetics of these relationships here, and then these feature predictions in the wild, and then looking at stabilizing selection, where the extremes are actually harmful, but phenotypes are moving towards average. Meyer with allopatric species separation, where there's reproductive isolation that leads to species that are initially compatible no longer being able to interbreed because they've been isolated for so long that inversions or other chromosomal differences can in fact lead to speciation events. Simpson looking at paleontology not being linear, but in fact branching and irregular and non-directional, of sort of exploring this space. Stebbings looking at plant polyploidy as a barrier and instantaneous creation of new species when you have a duplication event of the genome, for example, that leads to no longer compatible species with each other. Williams raising up the selfish gene hypothesis, and Lewis with the Hox genes and the evolution of the body plan and this deep homology in embryo versus ancestral species, and this whole Evo-Devo relationship. Kimura building these molecular clocks and this coevolution and this arms race of sexual reproduction. Then sociobiology and punctuated equilibrium. And then later, this concept of exaptation, of basically reusing things in evolution that are initially evolved for different functions, learned behaviors and this plasticity, or the battle between maternal and paternal genomes that are leading to either maternal and paternal effects through imprinting. And then, of course, the advent of whole genome sequencing and phylogenetics and phylogenomics and epigenomics and epigenetic inheritance, the construction of niches through evolution and this coevolution between the genes and the environments that they shape. And then the concept of evolvability, of basically getting better at evolving and being able to go back through previous forms by maintaining this ancestral diversity. Gene transfer and phylogenetic networks rather than sort of traditional bifurcating phylogenies. And again, the concept of endosymbiosis, of being able to engulf another species and live within it, or have it live within you. Hybridization and evolutionary psychology, mate selection, sexual selection, moving from genes to organisms and ultimately ecology level evolution. And again, Kunin proposing this post-mortem synthesis, incorporating all these ideas. So anyway, I just want to expose you to all of these ideas. There's a tremendously rich literature. We don't have, of course, the opportunity to dive into all of that. But in fact, I encourage you to take, for example, Bob Berwick's course on evolution next term, and other courses on evolution. Anyway, that's the lecture for today. Who feels that they've learned stuff today? Lovely. So we're at 70%, 30%, 0, 0, 0. Okay. Thanks, everyone. And that's basically all the material for the exam. Next time's lecture will be on single-cell genomics, and this will not be covered in the exam, but I still encourage you to attend. Thanks, everyone.
Professor?
Bye. Yeah?